Yeah. Okay. Do you want to follow up on that, Eric?
Well, I'm sort of, inspired to think of one thing, and I don't think it's controversial, but when in working with Alka and Ravi with the consortium that they have put together and observing the growth, and then subsequently reading things like their NIH grant and the manuscripts that they put out, there is a belief amongst these scientists and clinicians that genome structure and variation of genome structure.
So welcome to this presentation of AGBT and Mendel's Pod Live, the future of genome analysis. Welcome to the AGBT audience and to our Mendel's Pod audience who will be watching this later in the week. I'm Farrell Timpson, which makes me very pleased to be interviewing two leaders of Bionano Genomics, the genome mapping company. Erik Holmlin, CEO, and Alka Chabi, CMO.
Innovation is not cheap, and so capital is required. But we're at a time where people who are putting capital to work are recognizing the potential that lies ahead in genomics, and so they're getting behind companies like Bionano and many others to really give us the fuel we need to drive the big biology. Genome structure and variation of genome structure is very likely to be as important, that's the sort of political way of saying it, and maybe even more important than sequence variation. And so why is that? Of course, I think that because we're the structural variation company. That's what it says there. We love structural variation. We don't focus on sequence variants. And there's a reason for that, because if you want to get to single base resolution, you have to slow the system down. So we made a conscious decision to say, well, we're not going to do single base resolution because we want to go fast. Right. It's got to be relevant for large studies, clinical use, and clinical applications of LDTs is what I mean to say. It's got to go fast. So we'll sacrifice the single base resolution. But the other thinking is that large rearrangements in the genome can impact many genes, many other functional elements in a single swath. So, like a tornado is a really big deal. And when it goes through a town, it's pretty mangled often afterwards, but a heavy windstorm doesn't often produce that effect. And so structural variation, the hypothesis is that it's driving a lot of biology. The technical challenge is that because of the repetitiveness of the human genome, it's not just the human genome, plant genomes are even more repetitive than the human genome. The human genome is the one that gets all the funding, good. But it's so repetitive that resolving the repetition is required to see its structure. And so, I believe that the work that they're doing in this consortium around COVID and, how patients respond to the disease and progress, these other areas of we talked about neurodegenerative disorders, cancer, the amount of biology that's laying there, that's been invisible. It used to be called the dark matter.
Right.
It's huge. It's big. It's substantially, substantially large. Maybe larger. That's not for me to say. That's for us to figure out.
Well, I appreciate you going into that. I mean, if somebody's going to know, you should have a kind of sense of that, right? With your customer base. And I understand that, it's kind of an intuitive sense here. You use the word, I believe. Because we're just nibbling at the edges, maybe. Do you have a sense of where we are in big biology? I mean, are we just at the beginning? Or are we kind of in the middle? Are you getting a sense of that?
Yeah, I mean, we have thought about it in the following way. And so that is, when we looked at the first 2 decades of this century, I think clearly the 2000s were the microarray, gene expression, SNPs, and and next generation sequencing came in, revolutionized Sanger sequencing, and brought genome analysis, especially DNA analysis, to an incredible level, industrialized it, right? Really made it something that could go affordably and fast, so you could get populations done. And to a certain degree, that has leveled off. If we look at the outcomes, though, they're extraordinary. So, I was in molecular diagnostics right out of school, and I observed Randy Scott and Genomic Health. And when we think about what Genomic Health did to create molecular diagnostics, deal with the reimbursement and all of those things, incredible, but enabled by microarrays and other genomics technologies.
Yeah, but they were
spawned
the
incredible
pioneer,
industry.
and they had to be the first and the first and the first and the first. Yeah,
The first is tough, but amazing. And now when we look across that industry, we see a diaspora of companies that grew out of that idea. And for those people who pay attention to the stock market, are very handsomely valued right now. So that whole industry came out of those first 2 decades. But look at adaptive biotechnologies and sequencing all of the T-cell receptors. This is something that NGS and genomics has driven. So but I do believe it's fair to say that now the focus is much more on content and applications of the technology. Those technologies are not as advancing at as fast a rate. Illumina has acquired Grail. They have an intense focus on content and application. So big biology is going to come somewhere else. We know about spatial biology. It's incredible. Single cell biology. But within the genome, within the nucleus of the cell, structural variation is it. So we feel we're very much at the beginning of the movie. We think it's going to be an incredible movie. And we're excited to be at the forefront in helping it unfold. But we're very much at the beginning. And that creates a unique challenge for scientists who want to work in this space, because when you do sequencing and you do resequencing and you find single nucleotide variants, there is an abundance of databases out there for which you can look for that variant and see if somebody else has seen it and described in the literature. There has been no tool that allows that systematic discovery and cataloging of structural variations. One of the most important steps in that process was taken last week by Charles Lee, Jan Corbell, Evan Eichler, who are the co-chairs of the 1,000 Genomes Consortium, which has been focusing on structural variation for over a decade. And they published a paper.
Character in this movie.
Big players, and they published an incredible paper. Bionano was part of it. Long read sequencing was a part of it. Illumina sequencing was a part of it. And the most complete collection of genome variants ever, spanning ethnicities. And this is the starting point. There's some 100,000 or so variants in there. And so this is really the start of having the information to begin to rationalize that N+1 discovery. And so when we build that infrastructure for discovery, which is ongoing, those guys have done it, but there are other consortia working at this. Ravi's consortia is looking in a narrow field, but pretty topical: COVID susceptibility and COVID response, host genome response. We'll build these databases, and that will enable this wave to build and build and build. And it seems like an incredible opportunity for discovery, and it seems like it has to be the area of big biology. And one thing that's unique about it, when you compare it back to the first 20 years of the century,
okay,
is that the era of structural variation is coming off a foundation of structural variation driving so much clinical practice already today. SNPs were not such a big deal. We're not such a big part of mainstream medicine, but today, if you have a genetic disease, you're getting chromosome microarrays. If you have cancer, you're going to be karyotyped. And so now these technologies will build on that foundation. So
that's true. There are some tracks that have already been laid, right? There are a lot of tracks.
Absolutely.
Yeah. Okay, so what's the path to actually get a test reimbursed? Like, how far away from that are you guys, and what do you have to do to get there?
Well, some of the laboratory developed tests that are running on Saphyr today are reimbursed today because they're looking for analytes for which there are existing CPT codes that are analyte specific. Other CPT codes are more technology specific, and so they apply to a specific legacy or incumbent technology, and maybe not so obviously apply to the Saphyr system. And so for those types of LDTs, and a good example is just genome-wide structural variation for a genetic disorder analysis. So the comparison would be chromosomal microarray. That process involves getting the data, doing the analytical validation, and we have some of that. There are a handful of papers have been published, and we're going to drive more and more of those papers. And so getting the analytical validation, I mean, people appreciate it when I make claims about the capabilities of our technology, but they appreciate it more when we prove it in a publication. So we need to do that. And it's not just one paper, it's multiple papers. And it's also papers here that are coming from, US based institutions, because there are idiosyncrasies associated with those genomes and those samples and those programs. So a data set, and we're working on that, but it's not just a data set, it's really working with other labs or having other labs see the need for and utility for the capability, setting it up, developing LDTs, applying for their own reimbursement codes, proprietary laboratory analysis codes, and then testing patients and sending the bills to insurance companies. Because when that happens, it's analogous to what happens in the research side of things. The first person who sends a grant to the NIH hears that nobody's at the NIH has ever heard of that. Can't be any good. But when they're, which is why I left academics, but I'm sorry, God bless all these AGBT folks who are
Erik was an
fighting it out. I was, so if there is a critical mass of adoption in the field, then it starts to make sense for payers to pay more universally. Right. And so that's the path that we're on. Right. And that the market is on.
Yeah. You mentioned Genomic Health. I mean, they were a lab who got reimbursement for their tests, but of course they weren't an instrument manufacturer. They were using other instruments. Right. So it'll be a lab like Greenwood Genetic Center, which will get using your instrument, which will get the test reimbursed, right?
Right.
Yeah.
So,
and the distinction, I would say, I mean, the distinction is pretty important, right? Because, Genomic Health, looking at certain patients within, breast cancer stage 2, looking at a set of biomarkers, which based on a panel that they have curated and looking at patient outcomes and working very hard to determine what is the right way of applying that information, adjuvant chemotherapy or not, and how does that improve outcomes in the long run. So establishing this idea of clinical validity and clinical utility. We don't contend with that, because the tests that our end users run on the Saphyr when they develop an LDT are the tests that they are running on karyotyping, right? So the clinical validity and utility is already established, and that is not something that needs to be argued about. What needs to be established is that the performance is comparable to those existing methods or better, and that there's a compelling reason to switch. And so that's what we focus our efforts on.
So Erik, let's end and look forward with you. Our last question, we were talking about reimbursement, made me think the other tools company, the sequencing tools companies are becoming diagnostics companies. Actually, they are seeking FDA approvals, and Illumina. You just talked about Illumina buying Grail. Illumina's turned into a diagnostics company. I'm curious, for you to look out long term at your future, but also short term. How are you going to spend that $400 million? I'm sure you have customers in the audience. What can they expect over the next year as well?
Sure. So, I think that our focus on performance of optical genome mapping in the Sapphire system is really kind of everything for us, and that's been kind of proven over the years. The faster we make the system, the easier it is to use, the more affordable it is, the more people there are out there who are able to adopt it and want to adopt it. So it's continuing to focus on that. So a dimension that Alka brings is this ability to do these types of studies outside, bring third-party unbiased data into the picture. And so now we have product improvement going along with sort of product development, and product improvement going on with market development to define the applications, connect the dots for end users of where and how to use the technology. So it's really a really good alignment of the outside and the inside of what we can do. To be very specific, we want to continue to increase the throughput of the system, and so we've talked about this. We achieved a level of about 5,000 human genomes per year. When you do cancer or you go really deep in the genome, those numbers go down a little bit, and so we just want to keep doubling down on that. That was a 14-fold improvement from the time that we launched the Sapphire system at AGBT. Lisa, I saw you earlier coming on, so thank you for the venue, as always, to be able to showcase the capabilities. But we launched that at AGBT, 364 human genomes per year, which was something that was incredible. It's now up to 5,000, 14-fold improvement. And so we're sort of going for another 14-fold. Now that's going to take place over a period of years, but we will focus on that. And so people who are adopting technology today will enjoy additional improvements based on software and other capabilities that we bring in. Cost is something that we're very focused on. And so we want to continue to improve our own manufacturing methods so that the cost of manufacturing the product can go down continuously. It has been, but it will continue to go down. And we sort of get it. Like, we're not going to keep all those savings for ourselves. We're going to pass them on to the market, because we see that when these assays get more and more affordable, there's more and more adoption. So those are things that we're focusing on. So I hope people don't hear this and wait to buy the system or buy the product because, oh, I'll wait till it gets cheaper. Don't do that. Start working now, and then when the prices go down, your volume can go up. You can do 2 times more patients and so forth in your study. So those are some really important, I think, blocking and tackling initiatives along with the studies that Alka is going for. But we raised I don't know how else to call it. I mean, it's a ton of cash. We've always been capital starved. This has been a limitation of Bionano Genomics from the time that I joined. I have been the CEO for over 10 years now. I joined in January of 2011. And I didn't have any gray hairs when I joined.
Okay, so that
my wife
scaled up
said
nicely.
to me the other day, my wife the other day says, I only have gray hairs now. So, there's the natural progression, but we view it as having another decade to work on this, and that it's our calling. It's our calling to drive this forward. And so, are we going to become more of a diagnostics company? Guess what? In August of last year, we acquired a CLIA business focused on pediatric neurodevelopmental disorders. So we are selling services. It's called Lineagen. So through that business, we sell services. We have salespeople who call on pediatric neurodevelopmental specialists and pediatric genetics specialists every single day. We have tens of samples coming in the door, hundreds of samples per month of kids who are trying, and their families who are trying to figure out what is driving their biology by looking at their genome. And the reason that we did that, and maybe this is what you were talking about, is that if you want to be good at providing solutions that help diagnosing patients, you got to know something about diagnosing patients. If you want payers to pay for a test that your customers are going to be buying from you and utilizing, you ought to be talking to payers. And so through the acquisition of Lineagen, which is an incredible team based in Salt Lake City and around the country, and the sales side, we have a whole host of contracts with payers. And so we're interacting with them multiple times per day, right? It's part of our business. We're interacting with patients. We have genetic counselors. We're explaining to families what their chromosomal microarray and whole exome sequencing results mean. So we have that. And so when I think about that in the future, what I think about is when we integrate Saphyr into that business, and now you have Saphyr surrounded by whole genome sequencing, whole exome sequencing, and all of these tools. I actually don't know exactly what that's going to produce. I just know it's going to be amazing. The capabilities will be amazing. And by being fundamentally a life sciences instrumentation company, our goal is to proliferate the technology. And so we're going to figure these things out. And as you can see, we're talkative. We're going to tell everybody about it. Right? So our goal is to figure stuff out and then teach people how to leverage those tools and those capabilities. That gives us a ton of joy. That gives us a ton of motivation. We know it's all directed at helping people. And so that's why we look at the next decade and say, for us, it's the decade of Bionano, but it's the decade of structural variation analysis.
Well, it's really fun to share your passion for this, and I look forward to hearing more about that, seeing that in the future. Erik, Alka, this has been a real pleasure. Thank you.
Thank you very much.
Thank you, Daryl.
And thanks to the folks at AGBT and LabRoots for making this a successful presentation. Thank you, everyone.
Thank you.